Well, and now that we've walked through a number of these modes of inheritance, we've reached the sort of the last ones that we're going to learn about that are modes of inheritance. Now, these are going to be several different names that we're going to group together under one idea, and that idea is this idea of gene interactions or locus interactions. Either word will work. The basic idea is this, and when we talked about all of these other modes of inheritance, we concentrated primarily on the segregation approach, right? So we said you received one homolog from one parent, one from another, and the alleles that those two would interact. They might interact as a dominant, they might interact as a codominant, they might interact as a lethal or whatever, but they were two alleles at a particular locus. Now what we're going to talk about are multiple loci or multiple genes actually interacting. So if these are the two homologs with locus 1, and this is another homolog with locus 2, these two are going to interact to produce effect on a phenotype. Okay? And in the real world, it can be 2, 3, or 4 of these loci working together to give a particular trait. But we're going to just learn and talk about the basis of this based on just 2 loci that are going to interact. We're going to call this gene interactions or loci interactions. This series of lectures to be divided into multiple parts because I'd like to introduce the ideas we do before, work us through some of the types of modes of inheritance that you would expect and the ratios that define those. And then we'll come back and because this is fairly complicated, we'll work several problems. So we'll probably divide the problems into maybe 2 sections or so. So let's take an example and kind of go and illustrate what we're dealing with here. Okay. If we take a true breeding, these are bell peppers, or they're called sweet peppers in other countries, but just take these peppers, and we have a true breeding red and a true breeding cream, and we were to cross those, we would see that we end up with red, right? We would have a nice red color. This is early on, maybe starting to suggest we have a dominance thing going on, right? We have some situation where we have maybe red being dominant over cream or whatever, right? That's what you would expect from a simple Mendelian. Now, if we take those and move to the F2, okay, so remember that we're starting out with a red and a cream, and we're having a red show up. If we take those 2 red F1s and cross them, we would expect, if it's simple Mendelian, to maybe see a 3-to-1 red to cream or something like that, right? But what we actually get, it looks like this. We end up with the reds recovered, the green showing back up in the F2, and then these 2 colors that didn't exist at all in any of the parents, either in the F1 parents or in the original parentals. And we thought, that's sort of odd. Well, of course, we don't know much at all until we look at the ratios. And when we see the ratios appear, the ratios look something like this, right? We have a ratio of 9/16 of them are red, 3/16 of them are peach, 3/16 are orange, and 1/16 is cream. Well, a 9-3-3-1 ratio. But that's Mendelian in the sense that if we had a dihybrid cross here, a 9-3-3-1 is one you've probably learned as a primary ratio for a dihybrid system where both of the traits both are dominant and recessive. But we don't see red shorts and red longs and cream shorts and cream. We don't see 2 traits here. We simply see 1 trait, which is the color, and we see it in a 9-3-3-1. Yet this 9-3-3-1 is really critical. It tells us that these individuals up here were dihybrid heterozygotes, right? Because it adds up to 16 parts. The one cross that we've dealt with so far that produces 16 parts is a dihybrid double heterozygous cross. So that means this was a dihybrid het, this was a dihybrid het, and we got these parts. All right, so what's going on here is the question. What's going on here is this. We have what is known as epistasis, okay? Epistasis is defined as the interaction between 2 or more loci that work to produce a single trait, okay? Now, classically, this has been defined as an interference because often, and we'll see some examples of this, where it actually prevents the expression of the other. But in the real world and looking through real genetic traits, lots of genetic traits, I should say, not just necessarily real, it doesn't have to be where they mask each other. It can be that they cooperate to produce something and they form. Now, you might think about it, if it's one if it's one locus affecting multiple traits, we call that pleiotropy, right? That was something we did earlier. That's different. Here we have two or more loci focusing down on a single trait. That's epistasis. So don't get those two confused. How does it work? Well, let's go back to our little simple model that we've used for biochemical pathways. And we said that, basically what's happening is that there's a substrate. It's eventually being converted into a product, which we think of as a trait. A locus will, by its alleles, will contribute in various ways to that pathway, either again enzymatically or structurally or whatever. Well, now we just have two loci that are contributing to the same pathway, and we may have 3 or 4 or 5, but in our case, we're just worried about 2. And the ability of this second locus, B, to do whatever it's going to do depends on the success of the A locus as part of its contribution, because this is a downstream system, okay? That's at least one example of how epistasis might work. Well, let's go back and look at these peppers and see if we can put this together, okay? I said the red was true breeding, so we'll just make it wild type true breeding. Okay, red is actually a wild type for many of the peppers, for KSMs. And so we'll have these, one locus we'll call the Y locus, and the second locus we'll call the C locus. Really the Y locus is the yellowing locus, and the C locus is one for controlling the amount of color. This is a complete recessive for both of those, and it'll produce cream. Well, obviously when we cross these, we're going to get a big Y from here, a little y from here, big C from here, C plus, and a C from here. Okay? And that's exactly what we get. In the F1 generation, we have a dihybrid het. You could think of this again, if you're getting confusing you a little bit, big A, big B, little b. That's all it is, all right? Nothing else unusual. All right, so we know when we cross big A, big B, little b times big A, big B, little b, hopefully by now you automatically know that's going to give me 16 parts. Genotypically, it's going to give me a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. So 16 parts that we're going to combine up. Okay, and that's really what happens here. What happens here is we get this, right? It turns out that this Y plus is a dominant over the little y. So it doesn't matter whether you have two Y pluses or Y with a little y, you get this control as long as you've got a big C. So if you get a big Y and a big C, or plus Y plus C, you get red. If you get a plus Y but two little c's, it downgrades the color and you end up with what we call peach. If you get two little y's but you have a dominant C over here, you get what's called orange. And then finally, this cream or lack of color, it's almost a white, is really caused by having a complete recessive condition. So these look exactly like you've seen before. We see big A blank, big B blank, okay, big A blank, little b, little a, big B, and this should have a blank next to it, and then little a, little b, right? But again, we don't have red talls and cream shorts, or something. We have just one trait, color, and happens to have 4 different phenotypes that play a role. Okay, all right. That's the introduction to epistasis. We're going to talk about a lot of models that follow that, but the basic idea is that we're going to take two loci, okay, one here, completely separate chromosome from one over here, and when they produce their products, they're actually going to interact with each other to make one trait. In this case, it's just color, all right? So in the next lecture, what I'm going to do is I'm going to walk through some of the very common forms of epistasis, at least the ratios that you might find important, and certainly ones that from the book and other places, and will be helpful for working problems. So just kind of give you a big overview by pulling out some types of epistasis and talking about those in detail.